Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Agglutination versus precipitation. 14 key differences and examples. Table of contents. Agglutination definition. Precipitation definition. Key differences. Agglutination versus precipitation. Examples of agglutination. Examples of precipitation. Agglutination definition. Agglutination refers to the clumping of antigens with their respective antibodies. Agglutination reactions involve particulate test antigens conjugated to a carrier. Carriers can be artificial, e.g. latex or charcoal particles, or biological, e.g. red blood cells. When red blood cells are involved, the process is called hemagglutination. With white blood cells, it is termed leukoagglutination. These conjugated particles react with the patient's serum, which may or may not contain antibodies. Results are based on the visible clumps formed by antigen-antibody complex formation. The sensitivity and accuracy of agglutination depend on factors such as incubation, duration with the antibody, amount and strength of antigen conjugated to the carrier, test conditions, including pH and protein concentration. Agglutination is widely used in diagnostic tests due to its simplicity and quick process. These include latex agglutination, flocculation tests, direct bacterial agglutination, hemagglutination. Agglutination is useful for detecting bacterial antigens, aiding bacterial identification. A more advanced technique is agglutination PCR, where antibodies bind to antigen DNA conjugates, leading to agglutination. DNA strands are quantified using qPCR after ligating with antibodies. Precipitation definition. Precipitation involves the binding of soluble antigens to their specific antibodies under optimum conditions, forming an insoluble precipitate. This reaction forms a lattice of antigen-antibody complexes, which precipitate out of the solution. For precipitation, the antibody must be bivalent, while the antigen can be bivalent or polyvalent. Precipitation occurs in the zone of equivalence, where antigen and antibody concentrations are equal. Immunological techniques utilizing precipitation include immunodiffusion, electroimmunodiffusion. This principle is also applied in analytical chemistry for detecting functional groups in chemical substances. When lighter precipitates are formed, flocculation occurs, where the precipitate floats instead of sedimenting. Precipitation reactions are often performed on semi-solid surfaces, like agar media or cellulose acetate. Precipitates remain suspended until gravity causes sedimentation. Key differences between agglutination and precipitation. Let's explore the key differences between agglutination and precipitation in a clear and concise manner. Definition. Agglutination is the clumping of antigens with their respective antibodies, whereas precipitation involves the binding of soluble antigens with specific antibodies to form an insoluble precipitate. Antigen size. In agglutination, the antigens are smaller, while in precipitation, the antigens are relatively larger. Solubility. Agglutination works with insoluble antigens, while precipitation deals with soluble antigens. Sensitivity. Agglutination reactions are more sensitive compared to precipitation reactions. Principle. Agglutination is based on particle clumping, while precipitation relies on the formation of lattices or cross-linkages. Reactions involved. Agglutination involves complex forming chemical reactions, whereas precipitation involves reactions between ions and salts. Media requirement. Agglutination does not require a gel matrix, but precipitation often requires a liquid or semi-solid matrix. Resulting compound. Agglutination results in agglutinates, while precipitation forms precipitates. Nature of complex formed. Agglutinates typically settle at the bottom, while precipitants may remain suspended or settle slowly. Nature of reactants. Agglutination starts with particles, while precipitation begins with ions. Requirements. Agglutination is a surface reaction, requiring exposed antigen surfaces. Precipitation requires equal concentrations of antigen and antibody. Reaction. Time. Agglutination reactions complete in minutes to hours, while precipitation may take hours or even days. Appearance of end products. Agglutination produces large visible aggregates, whereas precipitation creates insoluble aggregates. Applications. Agglutination is commonly used in blood grouping, while precipitation is used for quantitative analysis and pigment formation. These differences highlight how agglutination and precipitation serve unique purposes in diagnostic and analytical settings. Thank you for listening. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Examples of agglutination. Hemagglutination assay. This diagnostic technique detects viruses, bacteria, and antibodies. Antigens from viruses or bacteria bind to sialic acid receptors on red blood cells, forming a lattice network. The lattice formation depends on the concentrations of antigens and RBCs. At low antigen concentrations, RBCs settle at the bottom without forming lattices. Hemagglutination mimics viral mechanisms during infections. A control well is used to compare antigen concentrations. Based on agglutination levels, the antigen and organism concentrations in the sample can be determined. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Examples of precipitation. Immunodiffusion precipitation test. This immunological technique detects and quantifies antibodies and antigens, typically immunoglobulins and nuclear antigens. Antigen and antibody are applied to adjacent wells. As they diffuse towards each other, precipitates form visible lines where interactions occur. Comparing multiple antigens in multiple wells is possible. Precipitation lines indicate the presence of specific antigens, enabling the detection of bacteria or viruses. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!